Okay, next topic is sort of a big one. It's sort of an important one these days. It is the immune system. So remember, one of the functions of your circulatory system is to manage your immune system, or to at least provide a network that transports leukocytes, white blood cells, which are part of the immune system, to all the areas of your body. It's where the security guards of your body patrol. So let's talk about your immune system a little bit in general, and then let's get into sort of the specific parts of the blood that have to do with the immune system. So your immune system, there are basically three levels of immunity that you have. The primary, which is called innate immunity, is a passive immune system. You're not doing anything. And that involves it's basically barriers, physical barriers that prevent pathogens from entering your body, entering your blood. So those physical barriers include, well, your skin is a big one, and this primary, it's not a response, but this primary level of defense, 99% of all pathogens it keeps out. It's like the doors are closed, no one can get in. Like I was saying, skin is one of them. You have dead skin cells. Your body is covered in dead skin. The layers underneath, there's many layers of dead skin. The layers underneath are the live skin, but you have dead skin on the outside. That's part of your immune response. The dead skin, because it's dead, it doesn't let anything infect it because it's dead. Bacteria can't grow in this kind of environment, not very well. There's nothing for them to eat. It's just this kind of hard, proteiny, dead skin. Viruses need live cells in order to be able to reproduce, so there's no cells for them to infect. And so there's nothing for bacteria to do. There's also, you secrete all kinds of oils all over your skin that have a low pH, that hurt bacteria, hurt viruses. You do have bacteria living on your skin. You have lots and lots, actually, of bacteria living on your skin, but they live passively. They are just simply, you are just the place where they live. They're not attacking you. It's sort of a symbiotic relationship because you provide them with a place to live. And what they do is they are also part of your passive immune system, is that these bacteria keep invaders out. They outcompete any invaders. Anything that lands on your skin, well, there's already bacteria there, and they're like, hey, screw off. This is our place. We're not harming this dude. Get out of here. And that's basically how that works. You have mucus, so you do have holes, right? You've got holes. These holes are dangerous. Things can get in through these holes. And so you have mucus in all of these holes. There's mucus. There's earwax in your ears, tears in your eyes, snot in your nose, saliva in your mouth. There are fluids and mucuses. So when pathogens or anything gets in, they are usually washed away by your tears, wash away pathogens from your eyes. There's also proteins and salts in your tears. That's why your tears taste salty. That will lyse any cells that they can produce. They're a very hypertonic environment for anything that lands on your eye. It's a super hypertonic environment, and therefore you'll have deplasmolysis of anything that lands on you. Mucus in your nose, stuff gets stuck in the mucus in your nose, and then you swallow that mucus and it goes into your stomach, and things are destroyed in your stomach acids. Your mouth, your saliva, it contains salivary amylase, as we learned, but it also contains other actually enzymes, and these particularly like to eat cell walls of bacterial cell walls. So you will dissolve bacterial cell walls in your mouth. It's very hard for bacteria to establish themselves there. There are bacteria that live in your mouth, of course, that can defeat these things. Bacteria are very versatile. But a lot of these things you swallow and they go into your stomach acid. The low pH of your stomach acid is pretty good at destroying things. You have your trachea, your upper respiratory tract has little cilia, little hairs and mucus on those hairs that are constantly in your trachea. They're constantly, so they're doing this. They're curled the other way, and they're pushing stuff up. up into your pharynx so that then it can be swallowed, so things don't actually make it into your lungs, or very few things. Most of the infections, note, that we get, coronavirus included, the flu, are of the respiratory tract. It's our very vulnerable area. We make it worse by smoking. Smoking marijuana, smoking cigarettes those cilia damage our ability to remove those things, those pathogens up and out so that we can swallow them and consume them. So those are some of the examples of passive immunity. They're physical barriers. That's the first line of defense. It's just the physical barriers that keep bacteria out. Oh, I was going to say hairs. I always like really hairs because we're hairy. We've got little tiny hairs, and what the hairs do are also in the nose and stuff. So a bacteria is floating along, it sees you, and it's floating towards you, and it's like, yes, I can finally infect this person, and it's getting there. It is. It can see the skin. Maybe there's a scratch on the skin. It's aiming for that scratch, but lo and behold, it gets stuck to a hair, and it never reaches the skin. There's the hair. It can see the skin, but it can't reach it because it's stuck in the hairs on your skin. So the hairs are part of your innate immunity as well. So that's the first line of defense that you have. And of course, this line can be breached. But like I said, 99%, nothing gets breached. But we've got some vulnerable zones. Respiratory tract, you get a cut, you get a scratch. Now your blood is out. Things can get in. Infections can get in. That's why we have another level, the secondary immune response. This is also an innate immune response. And the characteristics of this response is it's not specific to any kind of pathogens, not really. It's very general. It's very fast acting. And really what happens here, there are leukocytes here, white blood cells, that basically fall in the main group of them are phagocytes. So these are phagocytotic cells. Remember, from our cell membrane unit, we learned phagocytosis is when the cell membrane, it invagulates and pulls stuff in. So here, phagocytosis is these cells, they take invaders in, and then lysosomes, remember those endosomes that turn into lysosomes, fuse with that vesicle, and hydrolytic enzymes go in there and chop whatever it is that they've eaten out. And so the things that the secondary immune response, these phagocytes, can recognize is limited. They sort of have these receptors on the outside of them that in general terms are looking for things that they recognize that are pathogenic. So like the cell wall of bacteria, general things like that, some certain viral proteins, in general terms, and they're not specific to a specific bacteria, to a specific virus. When they recognize those things, they activate and they eat it. And as a result, because they're not specific, because they're very generalized, things can get past them. But if there's an infection, if there's bacteria set up, these will be working to eat, to consume, to phagocytose any invaders. The other thing that the innate immune response is responsible for is inflammation. So inflammation. Remember in this unit when we talked about capillary fluid exchange? So here's capillary fluid exchange. Here's the capillary and some fluid. If cells are damaged, these cells get damaged. And they're damaged for any reason. You fell down. You got burned, or a needle jabbed you, or bacteria or viruses are here and they're damaging these cells and breaking them open. When the cells break open, they release chemicals that signal the immune system and attract cells from this secondary immune response. And what happens is these certain type of cells called basophils, they arrive and they, under stimulation of these broken cell bits, let's say, will release a chemical called histamines. And what histamines do is they will make the walls of the capillary more permeable. They will actually open the walls of the capillary. The little openings will become more permeable. So the blood pressure will actually push more water out. There'll be a greater volume of water, and that leads to swelling. Plus, there's some other things. The cells, as they start to heat up, they're signaling for cells to heat up, and they're signaling that attracts phagocytes, those phagocytotic cells, those cells that eat stuff. The basophils will be released as well here, and they will just start eating things here, cleaning up. Basically, will be cleaning up the dead cell tissue. Number one, they'll be eating that. And number two, if there are any viruses or bacteria that are causing trouble here, they will be eating them as well. And this washing out, what it does is this, where the excess fluid now goes, it doesn't return back to the capillary and the venous end. A lot of it goes into the lymph, the lymphatic, and there's a unit, we'll be talking about the lymph shortly. And the lymphatic system is where lymphocytes live, and those are part of the tertiary, the third response. They're living there waiting to launch the third response. So things are being washed away down there, and leading to a tertiary response. So that's the secondary response. Is basically these phagocytotic cells that patrol the blood, patrol the intercellular fluid as well, patrol some of the mucous membranes and things like that. There's different types of these cells, and they phagocytose stuff that they generally recognize, but they're not specific, and they can be tricked. They can be evaded. So let's look at an example of so these cells, the secondary response. These are bad. So remember, all blood cells come from this multipotent hematopoietic stem cell, and they are all made in the bone marrow. And so these cells are particular. These are the phagocytotic cells. They have different sort of functions that they do. There's the basophil, that's the ones that releases histamines, and monocytes. These are the most sort of the most powerful phagocytotic cells. Monocytes, they differentiate into macrophages, which are these large cells that can engulf and eat large particles. And these other guys, they patrol other areas, the neutrophils and eosinophils. Eosinophils actually can phagocytose stuff, and they can release toxins that kill injured cells. Oh, before I forget, so yeah, there are also part of the secondary immune response are natural killer cells, NK cells. I don't know. These are somehow very popular these days. They're very rare, and how they work is so they if they feel that there's something wrong with a cell, so this could be a bacterial cell, but it could be your own cell. They're like, I think this cell's infected with a virus, because remember, viruses reproduce inside our cells. They're very hard to detect as a result, because they're inside our cells. They're hijacking our cells. The cell looks, for a while there, the cell looks normal. But natural killer cells, what they will do is they will release toxins that actually trigger a cell to kill itself. They're like, they talk cells into committing suicide. And so if there's viruses reproducing in that cell and these NK cells can detect it, they will tell those cells to kill themselves, so our own cells to kill themselves. And therefore prevent all that virus from multiplying. And again, these recognize general sort of things. They're not specific, okay? They're not. None of these are very specific. So, so we've beaten the primary response. We've beaten the secondary response. And so now, if we go here, so what's going on is. Say, virus has infected these cells, and now these cells are dying. It's attracted basophils, which have attracted, so we're still in the secondary response. And the secondary response blends into the tertiary response. And now the basophils have released histamines. We're getting a lot of stuff is washing out here, is being washed away. And but these cells are still dying. They're still infected by this virus bacteria. The infection is continuing on. There's more and more and more cell death. So more and more basophils are being attracted, more and more. This inflammation is also heating up that leads to fever. And these phagocytes are coming, these monocytes are coming, and they differentiate, they turn into macrophages. And these macrophages are eating everything in this area, cleaning out. And they're also consuming, the pathogen, because there'll be bacteria, there'll be virus particles here breaking out of these. And so they're consuming the virus particles, chopping them up. And then what macrophages do, they're sort of like serial killers. You know how serial killers like trophies and they display their trophies? I don't know. So what these macrophages do is they chop up the pathogen, they chop it up, and then they display the bits on their cell membrane. And they're like, look what I have done. I have committed murder. And they get washed away, and they get washed away into the lymphatic system. And in the lymphatic system are lymphocytes. So here are lymphocytes. And there's two types of lymphocytes. There are T cells and B cells. T cells, so B cells are all these cells are made in bone marrow, but B cells mature in the bone marrow. So they're done, they're finished their maturation in the bone marrow, then they enter the lymph. T cells don't mature in the bone marrow, they mature in the thymus, which is an organ of the lymphatic system. So they're called T cells. So you know that B cells stands for bone marrow, T cells, thymus. They mature in the thymus. So it just means where they mature. And there's two types of T cells. There are helper T cells, which there are a few different types of that, and cytotoxic T cells. And T cells participate in something called cell-mediated immunity. So in order for T cells to work, they have to make direct contact with a cell for them to work. And so remember, those macrophages have eaten the pathogen and are displaying the pathogen, and now they're in the lymphatic system, they're in, they're going to, there's certain organs in the lymphatic system like tonsils, lymph nodes. And there's also going to be some T cells around. It's not important where this is happening, but anyways, what happens is these T cells, they interact with these macrophages that have eaten this stuff, and what they do is those bits that they're displaying, the T cells will learn what those bits look like. Will sort of learn. They will gain receptors on there, what those bits look like, and then what they will do is they will start to divide. They will divide and divide and divide first. Now, if they already recognize this, if this is the first time, but if it's the second time and they already recognize the thing, they will divide immediately. But it takes time. If it's the first time that they're encountering this pathogen, it takes time. It takes a lot of those macrophages, it takes a lot of cell death to trigger this tertiary response. And the reason for that is because this tertiary response takes up a lot of energy and it's dangerous. So these cells, what they're going to do is they're going to start to divide, and they're also going to then promote. They're going to like pass these receptors onto. They're going to differentiate to cytotoxic T cells, so they'll have the receptor that is specific now for the very shape of the pathogens of the virus, for example. And so what will happen is now these cytotoxic cells will be released, and any of that pathogen now that is floating through the body, those receptors will be able to recognize that shape, those cytotoxic T cells, and they will release toxins to kill those cells, to make them commit cell death. Also, any cells of your own body, so if it's a virus, that are displaying maybe those sometimes the cells will actually put the viral bits out on their surface as well. The cytotoxic T cells will bind to your own cells and start to destroy them as well. So it's dangerous. And the thing that makes this dangerous, because it overwhelms the pathogen, is because these cells clone themselves. So basically, they will divide and 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 divide. And so there becomes a huge number of these toxic cells that are now going after every single one of these pathogens, anything that displays any sort of bit that resembles that thing that those macrophages were eating and chopping up and displaying on their body. So it's sort of a race. So the bacteria are infecting, or the viruses are infecting the cells, and who's going to be faster at reproducing? And these cytotoxic, it takes a huge amount of energy from you. You feel weak when you're mounting this response, because so much of your energy is going to cell division to make these cytotoxic T cells. And as long as you have these cytotoxic T cells dividing, eventually they try to overwhelm. So so they're doing that. The cytotoxic T cells, they have to physically contact the pathogen and infected cells to kill them. Now, also there are B cells, and B cells mature in the bone marrow. They're also float in your lymph. And what happens to B cells is B cells produce antibodies with the help of these helper T cells. So helper T cells are like, hey, I have learned the shape of this evil pathogen. I'm going to make cytotoxic T cells. I'm going to show them the structure of these pathogens, or just a bit of this pathogen that will directly bind now. And I'm going to help B cells by helping them create these things called antibodies. These are antibodies. Antibodies are called immunoglobulins. And how antibodies work, you can see they've got a binding, I was pointing over here. They've got a binding site over here and a binding site over here. And so this binding site corresponds to that thing that those T cells have learned, the shape of whatever that is. And usually what these shapes are is they will be proteins on a viral protein. On a virus particle, there'll be proteins that stick out from the virus protein. So in the case of coronavirus, remember there's the spike protein that helps corona get into our cells. So that would be one of the things that, when the macrophage eats a coronavirus, it will display those spike proteins as well, because it has broken the macrophage up and is now displaying those spike proteins or whatever other proteins. That's a very recognizable specific protein to coronavirus, so it's a good one for your immune system to learn. So now these antibodies now are specific to the shape of that spike protein. And what these B cells will do, they'll become activated. B cells will become activated, and they will start to divide. They will clone and clone and clone and clone, and they will start producing antibodies. That when B cells become activated, they become plasma cells. They're called plasma cells. And their job of making antibodies overwhelms them, and they don't live for very long, these B cells, because they dedicate their entire existence to making tons and tons of antibodies. So now antibodies are going to be floating through your body. And what antibodies do, these things bind to that spike protein. Let's say if you made an antibody that's specific to that spike protein, and you've got two binding sites. So you can bind one virus particle over here and another virus particle over here. So here's an example. There's some antibodies binding, and they create this clump of particles, either bacteria or virus particles, and they will clump together. This is called agglutination. It's sort of like when we had blood clotting. It also uses proteins, immunoglobulins. Globulins. It also uses globulins, but it's immunoglobulins, and it's a kind of different process in terms of it's binding pathogens here instead of red blood cells together to make a clot. It's binding pathogens together, so that now these pathogens, they can't move. They're all stuck together. They can't physically infect other cells because they're stuck They're stuck. They're in this huge clump. Now, macrophages is another type of macrophage. So the part of that secondary immune response. This is like candy to them. Antibodies. They want to eat agglutinized antibodies. So basically these antibodies are signaling to the macrophage like, hey, over here, look, I've got some bad guy and I've got me. So the macrophages come and they just eat these clumps. So the tertiary response is this race of cloning B cells and T cells to overwhelm the pathogen. How long a pathogen gets before the tertiary response is activated determines how sick you're going to get. Because if coronavirus is a novel virus, so we've never seen it, we don't have antibodies for this, we don't have T cells that recognize coronavirus. So coronavirus infects your body. If it makes it past the secondary response, it's got tricks to get past the secondary response. It invades cells all over your body and starts dividing and creates a huge number of corona particles. Then when finally the tertiary response kicks in, that tertiary response has to be huge. Huge. You're going to have to clone billions of these cells. It's going to drain all your resources to make all these B cells and T cells. Plus, there's going to be inflammation all over the place where all of those virus particles are, and fever, a heavy fever. If the stimulation keeps coming, a cell death and cell death. Every time those particles burst out of a cell, you got more cell death, you get more fever, more heat, and it's just devastating. And people who are weak is the wrong word. They're not at their best. Maybe they're already fighting something. They already have a chronic illness. They can become overwhelmed. And that's usually older folks, right? Older folks are dealing already with chronic illnesses. And so they can become overwhelmed, and your own body basically, it will kill you. It's not really the coronavirus that's killing you, it's your body's overwhelming response that is killing you. Now, the other amazing ability of T and B cells, certain T and B cells, so once they've learned the shape, so let's say they've learned the shape of that spike protein, and they've overwhelmed all the corona now, all the corona is gone, and we've won. And some of these B cells and T cells will differentiate into memory T and B cells, which are long-lived cells that will live in your lymphatic system, in parts of it, maybe in your spleen or wherever, in parts of your lymphatic system, and some of them will patrol the blood. Just a small number of them, just a tiny amount of them. And then what will happen is next time you get coronavirus, now, these things are around that are specific to recognizing coronavirus. There's none of that latency period of recognizing, is this bad, is this good? The secondary immune response doesn't play. You skip right away to the tertiary response. So basically the result of that is that coronavirus doesn't have time to infect and replicate itself in your cells. A coronavirus particle encounters pretty much immediately a T cell or a B cell that remembers it, if the number of those T cells and B cells are high enough. And then what will happen is immediately those T cells and B cells will start to divide, will start the response. They will start to clone themselves, cytotoxic T cells, antibodies being released immediately, but this time the number of virus particles is so much smaller that race to overwhelm it, there's no race. We win right away. Boom, there's more. They remove all the virus particles. The stimulus ends. It's actually a positive feedback loop. With the virus particle makes more T cells. Once the virus particles are gone, the chain is broken, and those lymphocytes disappear. They're gone. They've done their job. They will be reabsorbed. They will pass away, and you may not even notice that you had the infection. Now, this, of course, is the basis of how vaccines work. What vaccines do is they train your immune system. What vaccines do by introducing those, like, for example, in the case of corona, the mRNA vaccine, what it does is you're injected with the mRNA vaccine. And if we go, let's say, you're injected here in the tissues. Now, I don't know exactly what's in the mRNA vaccine, so I'm gonna speculate. I'm gonna generalize. So we know what mRNA does, right? It codes for genes. So the mRNA vaccine, the Moderna and the Pfizer vaccine, what gene they code for is the spike protein of corona. So you're not getting corona, you're just the spike protein. So that mRNA, I assume, is taken into your cells. There's some method that it uses to get into your cells. And your cells start to make the spike protein. They make a whole bunch of spike protein. Now, I assume that also at the same time, and most viruses have this, and of course the anti-vaccine crowd latches onto this, is there is something in the vaccine that causes cell death. So now the mRNA for the spike protein is not going to be causing cell death. But there is something in it that causes cell death on a very small local level. Either a very poor virus that sometimes there is actually a virus in the vaccine, like an ape virus, a sinew virus, that badly infects. It's more akin to, it likes to infect chimpanzees, but our genetics are close to chimpanzees, so infects us, but not very well. So it just kills a few cells. It's very slow to reproduce. It doesn't work very well in humans. Or some toxin is in the vaccine that produces some cell death, because you need that cell death to attract those macrophages, those basophils, those things that start the secondary immune response, which then also will wash away and trigger a small tertiary response. That's what ends up happening. And so you've got these cells producing spike protein, and at the same time you've got thing that's killing other cells. Those macrophages, they don't know what's killing those cells. They'll just eat everything and display everything in the neighborhood on their surface. And if there's lots of spike protein that's made by your cells that's being displayed, they will display lots of that spike protein, a huge amount of that spike protein. And then those helper T cells, they're just learning whatever's in that area. They will learn what spike protein looks like and produce an immune response for spike protein. And then next time real corona shows up, you will have memory B and T cells that have already seen spike protein, already associated with cell death, and mount a response to corona before it can get you. That's how vaccines work for all viruses and stuff. I mean, mRNA vaccine is new, is novel. In the past, what's happened is we would produce the protein ourselves, like outside of the body. So we would make the spike protein and inject somebody with spike protein and something that causes cell death. We would inject them with we would engineer a virus or something to, an inert virus, like a virus that doesn't cause major disease in a person. We would engineer that virus to, in addition to being, the virus doing itself, also produce spike protein or bits of, whatever it is that we want to create a vaccine against. That's the sort of the old-fashioned way of making vaccines. And in class, I'll talk about some of the others because it's not relevant. I don't want to make the video too long. Blood types. Okay, we got to talk about blood types. So, blood types. There are. This video will be too long. There are 4 blood types: type O, type A, type B, type AB. And, well, actually, and then O negative, O positive, so there are eight blood types. And you can look at this this graph or this table here. So a person with type A blood, basically what those A and B and AB and O mean is those antigens. So those things that I should have been using that term all along here, but the macrophage that displays the things on its thing, what those things that it's displaying are called antigens. Okay. And antigens doesn't mean necessarily a bad thing. We have our own antigens. All the cells in our body produce antigens to display. And remember, there are those glycoproteins or glycolipids, those little chains of sugar. Often that's what those antigens are. Those little chains of sugar that are determined by our DNA. Now, blood cells are unique as everybody produces the same sort of antigen depending on what blood type you are. So if you're an A blood type, you produce the A antigen on the surface of your red blood cells. And as a result, you have antibodies for the B antigen. So if you're a blood type A, you express the A antigen on your red blood cells and you produce antibodies against blood type B. So what would happen is if you were injected with B-type blood, your antibodies for that B-type blood would bind to those red blood cells and start to agglutinate those red blood cells. You get clumps of red blood cells and they couldn't carry oxygen, they wouldn't work. And you would start to mount an immune response against those blood cells. And then macrophages would come and start eating those red blood cells. If you are type B, you produce type B antigens, and you have antibodies for type A. If you are type AB, you have the genetics to produce type A antigen and type B antigen, and you display both on the surface of your red blood cells. There's AB, I don't know if you can see it from that far away. And you have no antibodies against A or B, which means you can be injected with type A blood and you have no antibodies for it, so it'll be just fine in your body. Type A blood, you are the universal receiver. You can be injected with type B blood and it will be totally fine because you do not have antibodies for type B or for type A because you express type A or type B, so you're not going to make antibodies against your own self. That's an autoimmune disease, and autoimmune diseases are a whole thing, another thing. Now, if you are type O, you have the genetics to produce no antigens. The surface of your red blood cells is clean. But you do have antibodies against type A and type B. So you cannot receive blood from type A, type B, or type AB because your immune system will attack that blood immediately. The only blood type that you can receive is type O, is another person for type O. But you can donate to every other blood type. You are the universal donor. Now, there is also something called the Rh factor, and the Rh factor, quite simply, is just another antigen. So if you are Rh positive, so if you are blood type A positive, you express the A antigen, and you express the Rh factor antigen. So you will not have antibodies to Rh. So you can receive Rh negative and the type A negative and type A positive blood. If you are type A negative, you do not have the Rh factor, and therefore you do not express the Rh factor antigen, but you do have antibodies for the Rh. So receiving A positive blood would be bad for you. The rarest blood type is O negative. Only something like 2%. There's a North American distribution. This is probably old. What is it? O negative. 8%, something like that, like 1%, 8%, 8%, 4%, 4%. I don't know what that average works out to, 5%, something like that. Of the population. Now, I'm O negative, so I am the universal donor. I express no antigens on my red blood cells, but I carry antibodies for A, for B, and for Rh factor. So I'm always being called by Canadian Blood Services to donate, but unfortunately my high blood pressure makes it so I cannot, because I take medication to control that blood pressure, and we wouldn't want to give that blood. So, so be aware which blood types have which antigen and which antibodies, and what that means. All right, so that's blood types for you. There we go. We're done. That's a long topic. Sorry.